Hello, my name is John Bachman, and I'm going to give a short demo of a self-updating and self-testing model of COVID-19 drug and disease mechanisms built and hosted by a platform called Emma, developed by myself and my colleague Ben Gyori. We also have a poster presentation, so rather than repeat everything that's in that video, I'm going to give a brief introduction and background here and then focus on demoing the Emma web application and the COVID-19 model. So Emma stands for the Ecosystem of Machine-Maintained Models with Automated Analysis. And it's a platform that uses text mining and knowledge assembly to continuously update causal models with newly published mechanistic findings. So under the hood, it uses several existing bio-NLP systems to extract relations and then uses our Indra software to assemble these relations into a knowledge representation suitable for causal modeling. The key is that Emma doesn't stop at just collecting new facts, but also regularly tests models derived from these facts for their ability to explain empirical observations. So an empirical observation could be, for example, a known perturbation response relationship relevant to a signaling pathway. And this process helps to highlight the findings that are not only new, but also have a meaningful impact on the causal structure of the model. So let's go to the Emma website. It's at emmaa.indra.bio. And when you first go to the site, you'll see the Emma dashboard where there's a set of cards, each one representing a different model. The models in Emma vary significantly in size and scope. There are disease models, pathway models, even small manually curated mechanistic models. And each model has a button View on Index where you can look at the representation of that model in the index network sharing site and query it and interact with it in different ways. So the other button here is the details button. So if we go to the details for the COVID-19 model, we arrive at the model dashboard where we can get a summary of what's in the model. So as I described in my poster, the COVID-19 model is built from mechanistic interactions that are derived from text mining COVID-19 relevant publications and also relevant host virus protein-protein interaction maps derived from multiple sources. So here we get to see what we've gotten from all those different sources. We can see the frequency of different statement types in the model, the most frequently mentioned agents, the most supported statements in terms of their number of supporting evidence sentences, and here how the number of statements in the model has changed over time with the incorporation of new publications. So currently the model is at 169,000 relations and increasing daily. And finally at the bottom you can see the statements that are added in the latest update rendered back into simple natural language. So one thing to note is that anywhere in the Emma website that you see one of these natural language descriptions of a mechanism like estrogen inhibits mortality, for example, you can click on it and see the sentence level evidence for that relation. And further, you can, by clicking this pencil, curate it as correct or incorrect if you are logged in. So back on the model dashboard, if we scroll back up, we can also go over to the tests tab. And this is where we can get some insight into how this model behaves with respect to a set of empirical tests. So in Emma, a test is an assertion about a perturbation response relationship that we want to check the model against. And a test is satisfied if a causal network derived from the statements in the model has a path that links the perturbation and the response. But there are, as you can imagine, different ways of taking a set of high-level relations and using them to generate a causal network. So the way that we resolve this is we actually assemble each model into multiple different types of networks with different causal semantics and display the results for all of these. And this is actually best illustrated by one of the other models in Emma that's a model of the RAS pathway called the RAS machine. And for this model, we use 4 different network types from an unsigned directed graph to the influence map of an executable PISB model. And because these network types have different causal granularity, they offer the user a kind of tunable precision recall trade-off in exploring explanations. And for fairly detailed models like this RAS model, these paths in the more causally granular network representations can be quite mechanistic. So for example, for this test, for GTP-bound active HRAS activates ETS2, this is something we expect the model to be able to explain. If we click on this check mark, we can see the explanation, and it follows the canonical route from HRAS through RAF to MEC to ERK and then down to ETS2. and makes reference to specific activating phosphorylation sites along this path. So going back to the COVID-19 model, it's worth noting that a model can actually be checked against distinct test corpora. And you can look at the results from each one. So for the COVID-19 model, we have 2 different test corpora of drug response assertions coming from different sources. The first is a set we've manually curated from 4 different drug repurposing papers, and that one's called COVID-19 curated tests. And the second are drawn from the MITRE therapeutic information browser, and this is these are the COVID-19 MITRE tests, and we can get different results for each of these. So looking here at these curated tests, these are empirical hits from in vitro drug screens. So one thing that I should note here is that there's only 2 different model types for this model of the signed and unsigned graph. This is largely for technical reasons because the COVID-19 model is the largest model we have in EMMA, and these are the most computationally tractable, these 2 representations. And we can see how the test passage rate has changed over time with different updates of the system and the content of the model. And we can scroll down here, and here's where we can see the drug response assertions. They take the form of a particular drug inhibits a particular coronavirus, and whether that test was satisfied by the signed graph and the unsigned graph. And we can take a look at a few of these. So if we go down to this one here, this says chemostat inhibits severe acute respiratory syndrome coronavirus 2. We click on the link here, we can see the explanation, and reassuringly we find that the path linking chemostat to SARS-2 goes through the known target of chemostat, TMPRSS2, which is also the viral co-receptor. Looking at another one, we have perhaps a more interesting one is this one here. Procellaridin inhibits SARS-2. And this one actually has multiple steps. This one goes from, it asserts that procellaridin inhibits CCL5. We can actually see the evidence for that. That actually comes from the Comparative Toxicogenomics Database, or CTD. Okay, so that's the curated database. And then the link between CCL5 and JAK2, we can see here that there's evidence for that from text mining. And then finally, the link between JAK2 and SARS-2, we can see here that inhibition of JAK2 blocks SARS-CoV-2 viral entry. So the idea here is that this path is saying that procellaridin overall is inhibiting JAK2, which would inhibit viral entry. So those are some examples, and thank you for watching and listening, and I hope to hear from you at the meeting.